But now let's talk about what's in the cell. Okay. And again, the best way that I like to put it is think of it as a village, and you have this moat that goes around the village. You have a castle there that controls everything inside the village. That's your nucleus. We're going to have roads all going through there. That's something called the Endoplasmic Reticulum. There's certain ones that are going to give the powerhouses of the cell. There's the electric companies. That's going to be the mitochondria. Think of it like that, okay, as we go along here. So this is, again, another picture of a cell, and we're going to talk about these parts now. Now, inside the cell, we have the big thing that's going to be there is going to be the nucleus. And like I said, that's the one that's going to control everything that the cell does, kind of like Washington, D.C. for the United States, okay? But think of also what's around the cell. So between the nucleus and the cell membrane is going to be a lot of fluid and a lot of other what we're going to call organelles, these other things, ribosomes and other structures that we're going to talk about. It's basically everything from the castle in the village and everything outside until you get to the moat. That's what we call cytoplasm. Cytoplasm is all the material that's between the plasma membrane and the nucleus. Okay? The cytosol is the fluid that's there, but not the solid structures that we're going to talk about. But there's other things in there too. There's amino acids floating in that water. There's electrolytes floating in that water. Okay? And that's what we call cytosol. We also have these organelles, these solid pieces of structures that we're going to see. It's the machinery of the cell to make sure it does its things. The road work, the little houses inside the village, the blacksmith and the carpenter area that's going to make things in there, all right? Some of these structures will have the same membrane that you just saw on the plasma membrane, okay? Some of them will have two membranes around themselves, giving them an extra protection. The nucleus is a good example of that, all right? Like Washington, D.C., you have to have a gate and then another gate right in front of it because you need extra protection there. And then there's some that don't even have membranes on it. They're actually made up of two particles, two subunits. Ribosome is a good example of that, okay? Then we have this cytoskeleton, okay? Let me just, cytoskeleton is just what it sounds like. It's the skeleton of the cell. Okay? And the functions of this is that it's going to determine the shape of the cell, the structural support, organized contents that we're going to talk about in there, all right? And it's going to direct movement of substances and contributes to cell movement, should the cell be moving. Some cells might turn into amoebas, not turn them into amoebas, but have an amoeba-like movements. And this is what's going to help it do that. And what the cytoskeleton is, it's a series of rods that are all through the cell itself. We have three different types of these rods. We have microfilaments. A good example of this is actin. And you'll get to know these later on as we go about talking about this throughout the next few weeks. These will make up something called microvilli, which will increase surface area. They'll also form cilia and flagellum, like the little tail that you see on the end of a sperm. It'll help with muscle contractility, so that muscles can contract, and also will maintain cell shape. We have intermediate filaments, which is going to strengthen the nuclear membrane, also known as the nuclear envelope, and will also maintain the cell shape. There will also be microtubules that organize the organelles and will move the cilia and flagellum. These, the microfilaments, will make the flagellum and the cilia. This will move it. And it'll also take part in cell division, a process that we're going to learn about later today of mitosis. And it kind of looks like this. You have all these rods that are going inside the cell, and that's what's going on here. Okay? So it kind of makes the shape of the cell. Now we also have things called centrioles and spindle fibers. Okay? These are located in something we call the centrosome, and I'm going to show you a picture of this so it'll make sense to you. There's it's a specialized area that's on one part on the outside of the nucleus, but it's just lying on the outside of that nucleus. And it's made up of two what we call centrioles. The functions is that it's going to form the basis of the cilia and the flagellum, and before the cell is ready to divide, these things are going to move on opposite ends of the nucleus, create what we call spindle fibers, and it's going to help move the chromosomes that we're going to talk about to two different cells eventually. The centrioles are kind of put on in a 90-degree angle on each other. This is a centrosome made up of two centrioles that are crisscrossed with each other. I'm not going to go into detail of how these are actually made. That to me is biochemistry. Later on you'll learn about that if you want to. Okay? Cilia, we have non-motile cilia and we have motile cilia. Cilia is when you look at the top of a cell. This is a cell. We could have little hairs that go up like this. They're not really hairs, they're cilia. And they move, or they may not move. In the ear and in the nasal cavity, there's fluid in this area over here. And when the fluid moves in a certain direction, this cilia is going to bend in that direction. And this could be hooked up to a nerve. If the cilia move in that direction, it's going to send electrical impulses to the nerve, and that goes to a part of your brain that says, oh, you're moving in a certain direction because the cilia moved in that direction. This is where we'll see it in the ear, where it deals with balance. An example of this is if you keep your eyes closed and you're in the passenger's side in a car, you could tell if the driver is moving left or right, even without your eyes open. Okay? It's the way that the water is moving in your ears and moving the cilia. So in this case, the cilia don't move on their own. That's why I'm saying it's non-motile. It doesn't move on its own. But we do have cilia that does move on its own. And we have a good example of this is in the trachea, the windpipe. When you breathe in stuff and a lot of particles, if you're in a sawmill or something like that, or you're breathing in a lot of bacteria, it starts building up in here. And you start producing a lot more mucus. When that happens, the cilia kind of push the mucus up to your mouth, create the cough reflex, and you cough it out. We also see cilia in the fallopian tubes. And the fallopian tubes have the cilia inside there because it kind of pushes the egg towards the uterus. Well, hopefully the sperm is there if you want to get pregnant. It says, oh, come on, he's a good guy. Yeah, go over there, it's okay. You know? All right, so that's where you'll see it on there. All right, and it's kind of looking like this, but it does this kind of sweep kind of thing if they move. Whiplash kind of thing. So it could be mucus up here, and this cilia can push the mucus. The mucus is going to collect those dust particles, the bacteria, and when it gets pretty heavy up there, then the cilia can kind of try and push it that way so it doesn't go down further into where your air sacs are, where there's going to be gas exchange. So what happens here, let's talk about like in the throat area where the trachea is. You have the saline layer at the apex, meaning like at this area here, you're going to have saline, an aqueous, a water solution up here. There's mucus in there, but let me explain to you what's happening here. chloride pumps, and just bear with me, I'm going to show you what this is normal, what's happening here, and I'm going to talk about a disease that you've all probably heard about, but I'm going to put it together here. Chloride pumps are located at the apex area here. In other words, at the top area of these cells. Chloride is going to be pumped out there through ATP. It's going to go out there. Now, that's going to make outside the cell more negative. Chloride is a negative charge. Okay? That's not a natural thing, or that's not a homeostatic thing. So what's going to happen is, as chloride is going out there, sodium is going to go out there too. So the chloride pump is pumping the chloride out there, making it more negative on the outside. That's saying, whoa, something's not right here because it's getting more negative. So the brain says, I'm going to put more positive out there, and that'll counteract the charge. Does that make sense? Becomes back to what it was, neutral. Except there's a problem with that. When you're putting negative out there and more positive out there, you're putting it more concentrated out there, are you not? You're making it more concentrated. So as sodium's going out there, water is going to follow to try and dilute those ions outside. You catching on with me? Okay? Negative out there, not good. Put positive out there, it neutralizes it. But there's too much ions out there, we have to dilute that. So we're going to put water out there. Okay? It's a domino effect. Cilia can beat freely in the aqueous watery solution. Because water is now out there, the cilia can now move and push things along. There's some mucus in there, but the mucus collected the particles, but the cilia can't, it's like quicksand, it's like really thick, it can't really move in there to push it upwards. So at this point, we're going to put more watery out there so it can push it upwards. It's all collected, now put it out there. That's the way it normally works, and thank God you don't even have to think about it. You just take it for granted. That's what happens in your body. Well, is everybody on the same page with me? Now let's talk about cystic fibrosis. Okay? Cystic fibrosis, there's a defect with the chloride pumps. They're there, they're just not working properly. So now you're not going to put chloride out there. The mucus is still out there, but the chloride can't go out there. If the chloride is not going to go out there, there's no way sodium wants to go out there. And if there's no sodium going out there, there's no water that wants to go out there. So what's left out there is mucus, a thick mucus. And the cilia can't move that thick mucus. So now you've got bacteria and you've got dust particles that are going to stay in that area. Well, that mucus is a great reservoir, a great area for bacteria to grow in. Oh, they love that. Bring a kid to a candy store. That's what's happening over here. Okay? So now they're going to start getting a lot of infections. Okay? So cystic fibrosis is a hereditary disease in which the cells make chloride pumps, but they don't really do their job. They're not working. So the chloride pumps can't make the saline solution that's out there, and the cilia can't move. So therefore, they're going to have, not just in the lungs here, in the trachea, they're going to have it where all these chloride pumps are supposed to be working, in the pancreas and other places, and now they're going to be really thickened with this mucus, and it's not going to work well. Usually people with cystic fibrosis, they have diabetes because things are just not working well in the pancreas. They get thick mucus, they can't even digest things properly. So you get this inadequate digestion of nutrients and absorption of oxygen because it's all happening here. They usually die because of chronic respiratory infections because they can't cough this out properly. They do get respiratory therapy where they kind of teach the parents how to basically break up the mucus in their trachea by not pounding but shaking and breaking it up on the back and then teaching the child to cough that out. Well, it may be easy for an 8-year-old to know how to cough, but not, let's say, a 1-year-old. You see what I'm saying? Okay? So 1 in 20 Caucasians carry this gene. So this is something that's pretty prevalent. And we'll talk about, what's the chance of you getting this if you carry the gene. Problem is with this, life expectancy is usually 30 years old, and they usually die of respiratory infections. Okay? Questions about cystic fibrosis? Okay? Again, I'm not trying to make this into a medical school kind of thing. I'm trying to put diseases in here to emphasize, to give the normal stuff meaning. Instead of just memorizing what the chloride pumps do, you're going to forget about that 2 years or 3 years from now. But you'll remember cystic fibrosis having something to do with the chloride pumps. It gives the normal stuff meaning, gives it some example of that. Okay, flagellum. There's only one cell that has a flagellum in the human body, and you ladies don't have it. Okay? The sperm is the only thing. Okay? And that's going to make the sperm swim to the lovely egg inside you. Okay? All right, now let's talk about the organelles. The nucleus. Now, what I'm going to do here is very briefly go over the next couple slides, what each one does, very briefly, and then I have one slide for each one, and I'll spend more time talking about it. So the nucleus, more than one nucleus, we say that there's 2 nuclei, and some cells do have more than one nucleus. All right? Skeletal muscles have a lot of nuclei in there because it needs to make a lot of protein, so it needs a lot of people helping out as much as possible. So the nucleus here is going to control all the activities in the cell, and it's going to contain what we call deoxyribonucleic acid, or DNA for short. This is the map of you, and we'll talk about that later on. It's very protective of it. It never leaves the nucleus. We're going to have a double lock on it so that things can't get in there to destroy the DNA. We also have inside the nucleus something called a nucleolus, and we could have more than one nucleolus there, and we say that there's 2 or 3 nucleoli. Okay? So, again, keep an eye on the spelling because that would make a big difference for these 4 things. The nucleolus found in the nucleus is going to make what we call RNA, ribonucleic acid, and it's used to make proteins from the DNA, and we're going to talk about that later on. Ribosomes are outside in the cytoplasm, so it's outside the nucleus. These are more like your stores or the places that are going to make things. The blacksmith that's in the village, the carpenter that's in the village and stuff, they're going to make things. The ribosomes here are going to make proteins. And proteins do a lot of different things. When you get into the chemistry lecture, you'll see a lot of different things that it does. Okay? So it's a site where protein synthesis happens. Some of these ribosomes, and they're very small, they're little balls, they may be just free-floating in the cytoplasm, or they may be right on a road. And that road is called the endoplasmic reticulum. The endoplasmic reticulum is going to go from the nucleus, and it's roadways throughout the whole cytoplasm. Okay? And it's going to transport materials from one part of the cell to another. Golgi apparatus, also known as the Golgi complex. This is going to, after the protein is made, then the Golgi apparatus is going to package it and put it into a form that makes some sense. Okay? It's basically taken, if you were going to take all my PowerPoints and put them onto paper, Xerox them, now you're going to take them to Staples, and Staples is going to put holes in them and put spiral bound around there and put a cover and a back part. It's going to package it. The protein's already made. Your copies are already made. They're just going to make it better to handle, suitable for whatever you're going to use it for. Lysosomes are in your cells, and they are containing a lot of digestive enzymes, so that when a bacteria, if it comes inside, like phagocytosis, it brings a bacteria in there, and we got to destroy it. The lysosome is going to fuse with that bacteria, and the lysosome will open up and let all its digestive enzymes go right into the bacteria and destroy it. So this is the police of the village that we're talking about here. And at times, when the cell is ready to get too old and want to die, then the lysosome will release all its enzymes and do like a suicide, a kamikaze type thing, and will just make that cell self-destruct. Okay?
So Lysol is kills bacteria, virus, and that's what.
I guess that's probably where they get. Yeah, someone else asked me that too. That's probably what I'm thinking. Yeah, yeah. Sometimes the medications and stuff, it kind of makes sense. Like Robitussin, tussive means to cough. So Robitussin, I guess they kind of said Robitussin is a cough medicine because tussive, Tussive, means to cough. So a lot of these companies like to add those kind of things in there, and yeah, I would think Lysol does that. Mitochondria is the electrical company that's in the village. It's the one that's going to make the ATP. It's taking in the oxygen and the glucose from parts of your body, bringing it in here, and it's going to make ATP. Okay, so those are the organelles. A few other ones, but those are the major ones, and then we'll get into the other ones now. Okay? So now let's talk about a little bit more detail about each one of these things. The nucleus, what's in purple over here, pinkish, okay, it's going to contain the blueprints of you, i.e. DNA, okay? It's going to contain 2 phospholipid bilayers, not just one, there'll be actually 2 in there. Since we have 2, we refer to it as a nuclear envelope. Since it has such vital information that's in there, we need to make sure nothing really gets in there. And we're going to put a double door on there, so to say. We also have, well, the DNA is chromatin. Okay? I'll explain. Chromatin and chromosomes, they're the same thing. Okay? Best way I can explain this, I might as well just do it now. Let's say we have 46 volumes of a reference encyclopedia set in the library, okay? And they're all bound together, okay? They're easier to manage when they're bound together. I could put 3 or 4 of them in my arm over here, it's no problem. If we want to divide them, it's easier to just do it that way. So that's what we call chromosomes. The information is DNA. The information in the encyclopedia set that's written on all the pages, that's the DNA. But they're bound in books. That's chromosomes. And the only time it's bound in books in a cell is when it's time to divide. Otherwise, the way it is that they take out each page out of the book and they lay them down on the floor. Every page. This way, when I need to make a copy or want to read about something about Victorian roofs, I don't know, then I could look, I could scan the whole thing and say, oh, there it is. I don't have to flip through all the pages. When it's laid out like this, very thin, light can come through it if it wanted to, that's called chromatin. When it's loosely coiled like that. Okay? When it's tightly bound in books, it's called chromosomes. It doesn't matter. DNA in the books or DNA written over here, either way, it's still DNA. It's just how it's structured. Okay? So that's why I put chromatin, chromosomes. Most of the time it's chromatin. The only time it's chromosomes is when we're ready to divide, something called mitosis. We also have the nucleolus in here, and that's the site of making ribosomes. And like I said, the nucleus is going to dictate everything that cell is going to do.
Question. There's something called chromatid. Isn't that just an arm of the chromosome?
Yep, and I'm going to show you that. Yep, it's part of a chromosome. And we'll talk about that when we actually get into what DNA looks like, genetic material looks like. Okay? Some cells don't have a nucleus. A red blood cell doesn't have a nucleus. It did early on, but when it became a mature red blood cell, it's not. The reason why it doesn't have a nucleus is because you'll learn, but inside a red blood cell, it's loaded with oxygen, and oxygen has to be bound to a protein called hemoglobin. If you put a big, good old nucleus in there, it's occupying too much of the red blood cell. So if we knock off the nucleus, we could pack it a lot more with hemoglobin. So that's why a red blood cell has no hemoglobin, or has no nucleus. Okay. Some cells have more than one nucleus, and I mentioned the skeletal muscle. There's also something called an osteoclast. It's because it's got to do a lot of different jobs, more than the usual, so it needs extra help. It's like two countries working together, two Washington, D.C., so to say, working together. Okay? The ribosomes. The ribosomes are what's going to make proteins. It's made up of not phospholipid bilayer, or any phospholipid for that matter, no membrane, but it's actually made up of two particles, two subunits. Something we call a 60S, which is pretty big, and a 40S, which is pretty small. They kind of go on, they kind of come together like a clam would. Okay? Some of them are free, they're just floating all in the cytoplasm. Some of them are attached to endoplasmic reticulum. These are the things that are like your Staples company, they're the stores. Some of them are freestanding, some of them are part of, that are actually on the road. Some of them could be on Route 10, some of them could be just, you know, out there, you have to drive out there, it's not on the main roads or anything. And they will contain proteins and a certain kind of RNA called rRNA. We'll talk about the different types later. We also have other RNAs. mRNA is going to read coded genetic messages. And when we get into it after the break, we'll talk about those. Our ribosomes are going to assemble, AA is amino acids, and they're going to take amino acids and put them all together, and you're going to have proteins. So that's what ribosomes are going to do. They're going to take the amino acids and make proteins out of them. The endoplasmic reticulum is this interconnected roadways that's happening throughout the whole cell. Some of them will have these red dots on them. Those are called, those are the ribosomes. All right, and somehow they all connect to the nucleus, the endoplasmic reticulum. It's like any road in the United States. Somehow it's all connected to Washington, D.C. Even Alaska, I wouldn't say Hawaii, but even Alaska is somehow connected by roads that are going to lead to Washington, D.C. Right? Right? So that's the same way. All the roads, all the endoplasmic reticulum, somehow, God bless you, is connected to the endoplasmic reticulum. I'm sorry, to the nucleus. If the endoplasmic reticulum has ribosomes on it, we say it's rough endoplasmic reticulum, or rough ER. Okay? This is going to contain the ribosomes, and this is specifically going to make phospholipids and proteins. But some endoplasmic reticulum don't have ribosomes, so they won't have any connection with proteins. In fact, they're going to be associated with making lipids. And that's called smooth ER. It's not rough. There's no ribosomes on it. It's smooth. And those where it's going to be making lipids or fats. The Golgi Apparatus, also known as the Golgi Complex. They kind of look like stacked envelopes. And the protein is already made. But now it needs to be packaged. Like I said, you make Xerox copies of all my PowerPoints. But now you're going to take them to Staples, and you have to package it so it's easier to manage with. Put holes in them and put spiral bounds around them. Okay? And when it does this, it can do the fate of that protein could do a few different things. Okay? It starts off over here, goes to this envelope, that envelope, that envelope. It modifies that protein to what it needs to do until it pinches off the last one, and now it's in what we call a vesicle. That protein is inside this vesicle. And the vesicle can do it could be like 3 different things it could do. It could either just be a secretory vesicle that could be stored throughout the cell and used for a later use, or it can actually become part of the plasma membrane, or it can also become a lysosome itself. So it's kind of showing you here the Golgi apparatus. Here's one pathway that can come out later on. It can go like this, can become part of the membrane itself, or it can become a lysosome that will go in with and do whatever the lysosome needs to do. Lysosomes. again, these are going to have digestive enzymes, and it's going to release them should a bacteria come inside the cell and kill them. If the cell is getting too old, it'll do like a quote-unquote cell suicide or autolysis. And it's going to release its digestive enzymes and just break down its own cell. Tay-Sachs disease. Who here has heard of Tay-Sachs? About half the class. Good. Tay-Sachs is a genetic disease, and what it is that it's the people who have this, they don't have a certain lysosomal enzyme called hexoamidase. This is very common in the acidic Jew population. And what happens here is, and we'll get into this, we'll talk about this again when we get into the nervous system, but just briefly, because we're talking about lysosomes. What's happening here is that normally we have glycolipids that are in the brain, and they need to be broken down. This enzyme would break it down. But if you don't have this enzyme, then these glycolipids are going to accumulate. And when they get so much, they're going to press down on the nerves, and the nerves won't function. There's too much of it in there. So it's going to interfere with nerve conduction, and that person can't do certain things. It'll lead to blindness, loss of coordination, and eventually dementia. And unfortunately, they usually die by 5 years old. Okay? So I put that. We'll talk more about it when we get to the nervous system. Okay? Peroxisomes. Similar to lysosomes, they have enzymes, but there's only one type of enzyme that's in there. It's called catalase. Catalase is an enzyme that's used to break down hydrogen peroxide. Hydrogen peroxide is a byproduct metabolism, but it's lethal to the cell. So we have to get rid of that hydrogen peroxide. And what it does, it breaks the hydrogen peroxide, which is H2O2, into water and oxygen, which then we could use that stuff.
So does that mean that lysosomes are produced in the Golgi apparatus? Does that say like...
It's not produced, it packages it. The protein, which is the enzyme, is made by the ribosome.
Okay.
But then it gets handed off to the Golgi apparatus to package it, form it into a certain structure that we can actually use. Okay? Mitochondria. It's the powerhouse of the cells, the thing that's going to make ATP. Okay? And this one also has two membranes on it. There's an outer membrane, and then we have this undulated membrane that happens in here. This undulation is called cristae. This is going to produce the ATP that we need. Because ATP is so vital for us to survive with, the mitochondria is not going to wait for permission for the nucleus to tell it to make ATP. This needs to make constant ATP all the time. It doesn't need to wait for the nucleus to do that. So because of that, the mitochondria has its own DNA. If it needs to make ATP, if it needs it, to make it, there's a lot of enzymes in here. So if we need to make more enzymes to make ATP, it's not going to ask for the nucleus's approval for that. It has its own right to do that. There's certain things, state laws, they don't have to get approval from the president to do certain things. Does that make sense? Okay, same thing here. So they have their own DNA. We call it mitochondrial DNA, mtDNA. Okay? They can actually replicate themselves. They don't even need the approval from the nucleus to do that either. If you need more ATP, the mitochondria says, we need to make more ATP. I'm not going to wait for nucleus to tell us about that. I'm just getting an approval. I'm going to make 2 more mitochondria so we get the extra help. Okay? It's that vital in our bodies to do that. All the energy in our body is derived from ATP. And like I said, you need glucose and oxygen to make that. If you don't have glucose, you don't have oxygen, you're not going to make ATP. Okay? Question?
Isn't there like a theory that the mitochondria was like a bacteria that went into the cell? That's why it had the
What is it? A bacteria? Say it
Yeah,
again.
like it went into the cell.
I don't know. I never heard of it that way. It's an interesting hypothesis, but I never heard of mitochondria being a bacteria.
Oh, with the evolutionary theory.
Yeah.
Yeah, I don't know exactly what I was.
Yeah. Which I guess it could make somewhat sense if it has its own DNA. All right. So I guess through evolution. If that's true, I don't know about that, but if it is true, then it's something we've actually piggybacked off of, and we just utilize it for better ourselves. Okay. So we have this mitochondrial DNA. Okay. When a sperm enters the egg, the sperm's mitochondria gets destroyed. So the mitochondria from the sperm never get into the egg. Does that make sense? But the egg has its own mitochondria. So it has its own DNA from the egg. Okay? Therefore, only maternal mitochondrial DNA can get passed onto the next generation. The male's doesn't. Does that make sense? Okay? Now, there are mutations that happen. Mitochondrial DNA mutates more often than DNA, regular DNA that's in your nucleus. They produce or can produce rare hereditary diseases. One is this myopathy, a certain muscle disease, mitochondrial myopathy. And if dad has mitochondrial myopathy, he can't pass it to his child. Does that make sense? Mom has it, she can pass it. Okay? So mom can pass on DNA that's mitochondrial. Dad cannot. Put in it, Mom, you gave me something Dad never could. I gave this to my mother. She said, I don't understand it. She doesn't understand the Far Side jokes either. So, but you see, you guys understand it now. Okay? So that's what that is. So there's our cell. Okay? And in the other school, we have a model of the cell on here. So that's what that is. So at least you want to see what that looks like.